It's a process that could possibly repeat. And I say possibly because it's not always a guarantee. What I need us to understand initially is I need us to understand how there are certain phases of this cell cycle. So I'm going to name the phases real quick. I'm going to put a start sign. I'm going to write start right there. I'm going to start at this position. And I want to work in the clockwise fashion, or clockwise way. And this would be G1 phase, the first gap phase. This is S, the S phase. Here's G2, which is the second gap. Over here we have mitosis. And over here we have cytokinesis. Remember, this is the process where cells are able to reproduce. And let's just assume that we stop at the same location in which we start. So G1 is the first phase, G2 is the third phase, S is in the middle of that, and then you have mitosis cytokinesis. Now when you learn this as freshmen, I think your brain learns to associate that this is the mitosis chapter. Mitosis is cell splitting. And that's the first thing I want us to get out of our head, is that mitosis does not mean cell splitting. It doesn't mean it at all on any level. What mitosis is the event in which you are splitting up the DNA. And this is a case in which you have DNA split, you distribute it to the daughter cells. And so the way that this works out, what we're looking for here is we're looking for a case in which you have an original parent cell, and that parent cell splits into two. So if I was to time lapse this forward, there's a point in time where it looks something like this. You can see daughter cell 1 and daughter cell 2 starting to emerge. And then lastly, the two daughter cells that exist perfectly.
During mitosis, is there a chance for mutations? So like one of the daughter cells is different.
I wouldn't call it a mutation at that point.
What would you call it?
Distribution error.
Okay.
It's called non-disjunction, more specifically. So what we're looking at through here is, do you see how this one cell is becoming two? See how the cell's splitting? Do you see the cells splitting here? This actually describes the process of cytokinesis more appropriately. Cytokinesis is when the cells are splitting. Mitosis is just a DNA splitting. That's really hard to view on this level, because seeing the DNA in a cell this size would be quite impossible. But I want us to get that mitosis doesn't mean that my cells split up. Cytokinesis better means that my cells split up into two. Mitosis is what happened before this to get your DNA to split up. Good?
So mitosis is DNA split.
It's DNA distribution.
Oh.
It's like taking copied DNA. It's like, let's say that I had 23 pairs of shoes, and I wanted to clone myself, and I wanted my clone to have the same 23 pairs of shoes I had. Well, then I would need to clone my 23 pairs of shoes, and I would need to distribute them to the original me and then the cloned me. Does that make sense?
Yeah.
That shoe distribution moment, which is representing the DNA, that would be mitosis.
Oh.
So we want to certainly get that. Now, we want to also remember that the DNA that is in this original parent, that is the blueprint for that creature. You were once upon a time one cell. You have divided, your cells have divided countless times, and here you stand today at trillions of cells, but derived from one original cell. And like a McDonald's, your recipe book, your DNA, is the same in each cell, at each restaurant. And if that's the case, we have to have a process, this cell cycle, that ensures that the new daughter cells are genetically the exact same as the original one. Isn't that the foundation of like how small businesses or now large businesses like McDonald's or Whataburger or any of the like, that's how they operate? You don't go to like different McDonald's like on different streets and they have different menus. They're serving the same thing in each one. They're replicas. They're clones. Your cells are the same way. The term interphase. The term interphase basically refers to the process of prepping for mitosis and cytokinesis. It basically is made up of G1, S, and G2. So these three that you're seeing right through here, those are all interphase. And you can see that a cell spends a fair amount of time in this during this process in the interphase. Yes?
Wait, how much time roughly would this circle of your little graph?
It depends. For human cells, it's different. I would say probably 30 minutes at a minimum. For bacterial cells, and humans are probably more like way longer, if I had to guess. For bacteria, I know this because this is like the fun fact written in the book. It's like some bacteria, like maybe E. coli or something very closely related to E. coli, can do this entire process of starting and then stopping and having one cell become two in under 8 minutes. Which would explain why you can have one bad bacteria on the burrito and you let the burrito sit out for like overnight, and the next thing you know you have billions of bacteria, because it's an exponential regression. 1 begets 2, 2 begets 4, the next thing you know you have a ton after just a couple of generations. Cool. Okay, so let's ask ourselves a couple questions. The first question is, why would we do this? Why are we going to go through this process that would take our one cell and make 2 out of it? Seems like taking 1 and making 2 out of it is just more work. No one likes having more stuff they have to do. So why go for it? Do it? Okay, so to replicate the cell, to have another one. Think about the video, the kid with the boo-boo. This would be a replenishment situation we have. Growth. You were originally 1 cell, now you're trillions of cells, you continue to grow. Obviously you have to be doing this. I could say replenish or repair. Bacteria, though, they do this for reproduction. Of their species. We don't do this for reproduction of our species. You don't do the cell cycle and then end up with mitosis at the end to form 2 new daughter cells from 1 original parent cell. You don't do that to make a child. You do something called meiosis to make a child, right? You make gamete cells, the egg and the sperm, which is different than what this is. But bacteria don't use eggs and sperm, so this is the process that we see used for them. So you could view yourself as a walking, talking clone. The difference between your cells can't be the menu, can't be the DNA. It's got to be which part of the menu that you're reading. Skin cells are reading skin-based DNA, whereas cells of the pancreas are reading DNA for pancreatic issues, pancreatic needs. It's like cloning libraries and sticking libraries in all different schools, but some schools are reading some books, some schools are reading different books than that. Okay. Let's talk a little bit about this business of chromosomes. DNA chromosomes. Because I think this issue, or I think this terminology becomes an issue sometimes early on. We certainly want to remember that the overall goal of the cell cycle is to produce two identical, and that's from a DNA perspective, two identical daughter cells. Want to produce two identical daughter cells. Now, if we want to produce two identical daughter cells, then we have to have a process that ensures that I can distribute DNA to both of them. Now, let's think about this. We're taking DNA from one cell, we're distributing it to two different cells that are both from that original parent. It would seem to me that just like moving items like from one house to another in the event that you move or you go to college maybe, you don't just randomly take the stapler to your car, the roll of tape to your car, a book to your car, then your one article of clothing to your car. What do? You package it. And so DNA is being as large as it is, it would be unwise to try to just move this string of DNA when you could just simply condense it down. Think about maybe yard work, if you've ever like at least watched someone do this. When I'm doing like the yard, I have like this large extension cord so I can like hook up some of my devices to it. When I'm done, I don't take this cord, all 100 feet of it, and just drag it behind me. I take it and I wind it up around my arm or do something like that. There's something about condensing it that makes it just more mobile. I can ensure that I get the entire cord to its like end spot or its begin spot. So we need to talk about how chromosomes relate to DNA. And the first thing that I want to focus on is what a chromosome is. It's a structure made of chromatin. I've kind of answered my question with another question because we're left with this idea of what is chromatin then. Chromosomes are made of chromatin, right, Patrick? So the question would be, what's chromatin? Well, chromatin is basically going to be a mixture of DNA and protein. And what those proteins can do is those proteins can basically allow the DNA to wind up around the proteins. Going back real quick, and this is going back to the video, I want to show you something. This is the video we were watching a while ago.
Cells. Outside the nucleus, sides of the cell, microtubule spindle. A forms loops.
Okay,
Nucleus,
like right
the
through
deep
here.
spindle.
You've got this DNA thing going on here, right? Yes?
Yes.
Bubble grows until it merges with an identical copies called proteins called histones.
And what they're showing you here is how that DNA beforehand had wound around these proteins called histones. And we'll talk more about histones as the class moves forward. But one thing you'll notice is you see this DNA circle, this loop, you know, if you will, that I'm going through. That's DNA. There's protein that is winding it up. Now, as the video moves forward,
Wait, I thought
The sister
the pink purple thing was DNA.
the pink thing is going to be the protein. This line, this string is the DNA. Do you see the DNA, how it's wrapping around this protein, and then there's DNA wrapping around this protein? Then it all coalesces, and you're left with this much larger structure, right?
Tight helical fibers.
Yes? Now, here we've got a chromosome. That was a chromosome. So, what chromatin is basically when you have packaged DNA and protein together. So, chromosomes are made of chromatin. Chromatin is the DNA and protein that has come together. Good? Now, let's talk about chromosomes. You have them, obviously. How many do you have? You have 46 chromosomes. That's like me saying that I have 46 shoes. So you have 23 pairs of chromosomes. Exactly.
Are they like exactly the same, like each pair, like?
They are different. They are different.
Okay.
And when we think about having these 23 pairs, each pair has a left foot and a right foot. Agreed?
Correct.
Okay. So I have 23 lefties, 23 righties, and together I have 23 pairs of shoes for a grand total of 46 individual shoes. We totally understand this concept. Now, here's the deal. You have, let's say, 2 boots. I'm wearing my little boots right here. I will wear them every day till you guys leave this building. My little boots, there's 1 pair, right? I have 23 other pairs of shoes, or 22 other pairs of shoes at home. There's my 46. I got 1 boot from my dad and 1 boot from my mom. Now, I can't tell you if it's the left one or the right one, but I know one of them is from my dad and one of them is from my mom. So for each pair of chromosomes, for each chromosome pair, 1 is from the dad and 1 is from the mom. They would have to be different because if the left boot and the right boot were the same, then that would mean the left and right chromosome were the same. The mom and dad chromosome were the same. And how can mom and dad's chromosomes be identical?
I know that part, but if you have for instance, there's boots and sneakers. My right sneakers for my dad.
Can't do that because the chromosomes, they pair up in a way that that kind of prevents that from happening. Let me show you a picture real quick.
So all the right ones are from one, and all the left.
Yeah, no, I gotcha.
Do you always give out the same chromosome? Do you guys give out the same shoe?
No, that's the deal. Is like for position 1, maybe maybe you got the dad's chromosome for the left part of it, and then mom's chromosome for the right, and then the next time it's different.
Oh.
And so what the reason, and that's more important for meiosis because that's what allows parents to have different kids, because typically people have more than 1 child, right? When they have more than 1 child, the children are never identical. And that right there is kind of feeding that principle that I could have like 5 kids if I wanted to one day, but the 5 kids I have, they wouldn't be identical. To help us through this, I'm going to show you guys a picture. This is off of the YouTube, or Google, I'm sorry. But do you see how I have these 23 positions here, 23 pairs? Maybe this chromosome right here is from the dad and this one's from the mom. The dad, the mom, the dad, whatever. Definitely, though, this is a boy, XY down at the bottom. Got the Y from the dad, the X from the mom. Cool? If it had been XX, it would have been a girl. But do you see how you have these pairs that seemingly kind of line up? Do you see how they kind of have the same size a little bit? The 2 at 4 look about the same. The 2 at 10 look about the same. Now that's about each time, but not every time. Do we see that? So these are pairs. Are they identical? Should these 2 chromosomes be the same?
No.
Because mom and dad shouldn't be twins.
That
Definitely.
would be impossible.
That would be weird.
Because you can't have a male and a female identical twin.
Shouldn't, no.
Well, could that ever happen? Ever? No.
No.
Yeah, I know that. What if it's like a little?
What if an identical twin has a child together?
They can't. No.
They
have
can't.
kids with identical twins. They would be
No.
no.
What if a parent had an identical twin of a boy and a girl?
That
It's
can't
impossible.
happen.
It can't happen.
No,
No.
because we have the same chromosomes. So ours look.
There's no, they would have to be both
Yeah.
X and X. You can have an XY and then an XX
So they
identical
have
twin set.
to determine.
Right. So, but even still, the example would be that the 2 chromosomes you see at position 11 can't be the same, because mom and dad shouldn't be the same entity, person, whatever you want to call it.
So can I just ask a question? So
Sure.
When this person has kids, their child will have one of those chromosomes.
Exactly. So you see how if you were to count up here, do you know how many little lines you'd count? 46. 23 times 2, 46. When you make a baby one day, how many chromosomes do you want your child to have?
23 from one person.
Yeah, exactly. 46 total, 23 from a mom, 23 from a dad. So do you see how you have, let's say this was me. You see how I have 1, 2 through X and Y? I'm going to give my kid one of these 2 from the 1, one of these 2 from the 2, one of these 2 from the 3. Hey, you know where I got this one from? My dad and my mom. So my kid is like half a mixture of my 2 parents. Like, how inventive.
So
You're the no-no.
Because my kid will come out and be like 25% of Jamie's mom, her dad, 25% of my dad, 25% of my mom. The kid's a quarter of each of the grandparents. The kid doesn't look like a hybrid of like your mom and your dad, because if that was the case, then your own child would be your brother or sister, essentially. That just never happens.
I know what you mean, but like, is it? This is just random now. But is it possible that like one child happens to get all of like from her father?
It could happen.
Girl, yeah, she gets just only her father's chromosomes.
But the chance of it happening would be, let's see, what is that? 2 to the power of 23.
So it's not very
So that those stats are like so extremely high. Like it would be never ever probably that you would get just exclusively. And there's also some other things that are going on, and we'll talk about this when we get to the reproduction phase of the class. We'll see that there are mechanisms that prevent even that from happening as a guarantee,
okay.
which means that we would all come out for the most part different.
I have a quick question. Why is the X chromosome significantly longer than the Y?
That I think there's more. Honestly, there's just more coding that's involved in the X versus the Y.
Okay.
Now, why did evolution take us there? I couldn't answer that question.
Because girls are more complicated.
That's the way I always remember it, but I didn't say that. But that is the way I remember it. Are we okay, though, with this idea of chromosomes, right? So let's talk about what's happening in some of your phases. Before we do that, though, there's some other terms that I want to kind of carry through or get through. The first is somatic cell, which is going to be a non-reproductive cell. There is a gamete cell, which is going to be a reproductive cell. We could say that the somatic cell is typically diploid, or 2N, whereas the gamete cell is typically haploid, formed by meiosis with the designation of N, where N equals 23 for humans, which means that there's 46 chromosomes that are unique in the somatic cell of a human, where there are just N, which is 23 chromosomes in a haploid cell, in a gamete cell specifically within a human. Different animals have different number of chromosomes. We're talking about 46 and 23 because we're humans. I think we'd like to keep it in that level.
Usually have less or more.
It's so different. Some creatures, you would think that since we're the most evolved, we might have the most, but it's completely false. Some things that are very simple have very few, definitely. But there's some things that are way less complicated than us, and they have way more than us. So I don't remember the exact examples, and I couldn't tell you a reason for that. I don't even care to guess. I have no idea. But it's at least nuts to think about, because you would think the more stuff you have, the more evolved you are, and it's actually not the case. So that's at least fun enough to explain. Let's talk about what's going on in like some of our pieces real quick. Let's talk about what's going on in the G1 phase. In the G1 phase, that's a part of interphase, we're basically prepping for the S phase. This is also a time of cell growth. It's also a time for organelle replication. So the G1 phase is all about growing up, prepping for this next phase, which is the S phase. And S phase will make so much more sense here in probably about 2 months, but for now, we need to know S phase is standing for the synthesis phase. Chapter 16 in your book takes apart how that entire process happens, from the listing of enzymes, overall process, but I want us to know this at least S stands for the synthesis phase, and that's of new DNA. Because if you think about it, if you're going to go from 1 cell to 2, you've got to have enough DNA to facilitate that need. That's just the logic of it. So the S phase is going to take care of having more DNA. And let me show you what's happening here. I'll try to do this like in time lapse with the markers, so hopefully we're all like in the zone with this. I don't know why I put these two so far apart. I'm sorry. You see what the dot 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 means? That means there's more stuff in between. There's the 23rd pair. How many pairs of chromosomes are you seeing here? I realize you only see 3, but you're assumed to think that there's 23, right?
Yeah.
Which is a grand total of how many chromosomes?
46.
And now I want, let's say this was original, like, let's put a circle around this and say this was my original parent cell. I'm going to form 2 daughter cells down here. How many chromosomes do I want each daughter cell to have? How many chromosomes do I want each daughter cells to have? 46. I want to clone this library, right? I want to clone the library. Now, if that's the case, then I need to clone these individual pieces. So here's what we're going to do. The S phase does what the green marker is about to do. Obviously, since this was the 23rd pair, this was the X. I'm sorry, this was the Y. This was the X. X's are larger. Cool. What has the green marker done? Replicated the chromosomes, right? So this is a chromosome and this is a chromosome. Now, when I point to this and I just said the word chromosome, did you hear me put an S at the end? No, which is singular. I'm keeping these two guys as singular. Why would I choose to do singular chromosome and point to this guy? Because there's obviously 2 pieces because they're the same thing. If I had a worksheet and I made a copy of it downstairs in the copy room, would I have worksheets then? Maybe, maybe not. I would say I don't because they're the same worksheet.
Would Auburn have twin? They're just like the same person.
Basically. Since it's saying twins, you should just be twin. We have a term here that describes the relationship between these two. The relationship between them is that these two are sister chromatids.
Sister Felix?
No questions. No, I'm just joking. What's up?
We have the same blue marker.
Oh, yay. Yeah. Sister chromatids. And what sister chromatids means is that it's basically copied chromosomes. Now these two appear to be attached, and they're attached at a certain spot called a centromere. So the centromere is the point at which the 2 sister chromatids are attached. Me?
Okay, so at this point, would it seem like they had, I guess, 92 from, like, it looks like...
They would look like they had 92, but you would still say technically they had 46 chromosomes because these two are the same. So 1, 2, 3, 4, 45, 46.
So it's just,
Even though there's 92 units in the cell.
right, okay.
The term
But-
chromosome and diploid and haploid only refers to genetic variability, not count of, like, or mass.
Okay, so basically mitosis isn't when it gets copied, that's when S phase is, and then when it's, like, mitosis is when the 2 chromatids split.
Exactly,
not
so...
when it's copied.
Right, so like we haven't even started mitosis yet. All I've gotten through is G1 and now I've gotten through the S phase.
Okay.
So the next thing that I want to go through is I want to actually go through what G2 is. And all we're talking about with G2 is we're talking about a prep for the mitosis. So again, this is like basically the final prep for mitosis. It's at this time that there's like you're ensuring the correct copying and all that other business. Like you're making sure, doing a double check, making sure that you're completely ready for mitosis.
What happens if you're not? Like, what if you really messed up? Would it just die?
Yeah, typically the cell just cancels the entire operation,
And
commits suicide, the lysosome splits open, it's over.
Oh, so that's why it goes so frequently, like why
Yeah, and this would explain, because if you think about it, let's think about this, and we're gonna get into more of this tomorrow. But when you're copying the DNA, what we're talking about here is we're talking about taking 6 billion letters essentially and rewriting them out. That's like someone taking that textbook of surgery essentially and rewriting it by hand. It would take you what? Maybe years to do that? Months? Not tomorrow, it would be done for sure. Take quite a bit of time. The enzyme in that case would be your hand. Now the enzyme on this level is called DNA polymerase, and we'll talk more about that enzyme later on, but I want us to understand that enzyme is pretty accurate in copying. It makes very few errors. It does it very quickly. Now with that being said, there would be errors along the way. Agreed? Nothing in this world could be perfect, not even the enzymes of biology could be perfect. And if that's the case, then there's a likelihood that you're going to copy DNA wrong every once in a while, which leads to the horrible sounding mutations that exist out there. Mutations, though, can be good, but are rarely good. They're never noticeably good. Most of the time mutations go and they occur and they are irrelevant because they're affecting an area of DNA that you never read. For instance, if I copied down a word wrong in the urinary system part of that textbook but no one was to ever read it, did I really make a copying error? It's like saying if a tree fell like in the rainforest but no one was around, does it make a noise? Does it?
Yes.
I guess, but is the noise of value to anyone? Would it ever scare anyone?
No.
Because there's no one there to scare. If I make a copying error in the urinary chapter and no one's ever going to read it, then the error goes unnoticed. And since most of your DNA codes for what we believe is nothing and it's just a waste of space, then that means that most errors, most mutation, most bad copies would go unnoticed because they're code for nothing. Now every once in a while they do cause an error, and hopefully at this point it recognizes that, because if it doesn't, that cell and the daughter cells maybe now have mutated DNA, and there could be a suffrage. The worst case, of course, would be the instance of cancer. Now, if there wasn't this unique checks and balance system involved in making sure that the copying went flawlessly, and that the double check to make sure it went flawlessly was there, you'd already all be dead. You'd already all be dead. But you're not. Who gets cancer 95 to 98% of the time? Young people or old people? Tell me about winning the lottery. I've never played the lottery. If I bought a lottery ticket today, do you suspect I would win today? If I bought a lottery ticket every day for 50 years, do you think I'd win after the 50th year probably? At least, right? The more times you play the game, the more likely you are to win the game. That's the simple stats behind it. So with that in mind, the more times your cells replicate, the more in danger you are of having that replication error that becomes cancer, which explains why older people get it. But fortunately, you have a lot of checks and balance systems that are helping to alleviate and making sure that this system goes flawlessly. Now, with that being said, there are some things you can be adding to your body or your body can encounter to make this event happen not so well and to prevent this check from happening perfectly. Some things would be like carcinogens or radiation or some other massive event. That's why you always have like nuclear areas are always a concern for people for this reason. Smoking is a concern. Smog is a concern. The amount of sun you get is a concern. All these events affect how effectively you can do this. Yes, ma'am.
So are you saying that the majority of DNA is just like irrelevant and we don't know what it's for?
Definitely. A fair amount of it is what we call junk DNA.
So if we just cut it out, we'd be okay? Probably not,
It's
but
kind of inefficient though, isn't it?
What?
To make all this stuff that's not even used.
Well, it is inefficient, but if you think about it, according to the theory of evolution, like we evolved from a lot of like prior creatures. And so like as you evolve and as you come up with new textbooks, and new technologies, still in the system there are the old technologies as well. Not used, not turned on, because the factors that turn them on aren't there. It's like saying that I have those keys. I'm not going to pull them out because it's such a hassle, but you've seen my key ring, right? I look like a janitor. Do you think I know what all those keys go to? No way. I use like 4 on average on a good day, and there's at least 5 keys on there that I couldn't tell you what they go to. But I'm afraid that one day, I'm going to encounter like that little door or whatever that needs that key. Do you understand what I'm saying? What I'm suggesting is that there is a lot of DNA that is junk, but as inefficient as it is to keep lugging around the extra keys, the extra DNA that is of your ancestors of a different species from billions of years ago, maybe, even though that seems pointless, with this mutation thing, it's actually not that bad, because it allows you to mess with, like, if you're going to mutate, you'd rather not have, like, only DNA that was coding for something of value, because when you mutated, you'd mutate something bad. Having a bunch of junk DNA kind of guarantees that you're going to mutate probably wasteful area, which means they go silently, which keeps you alive longer than 5 years. Cool? Awesome. To end today, I want to end on kind of a little bit of a note about mitosis. Let me see if I can't find my little picture. This is kind of what I'm looking for right here. This slide and the happenings of it is represented in your book. Now, I can't remember exactly what page it's on, but in chapter 12, this picture exists over the course of a left and right-hand side of a page. This is like the left-hand side of the page. I'm about to show you the picture that is on the right-hand page. It gives you this blown up, and it tells you in description right below that what's going on. And it is imperative, extremely imperative, that you understand this. I'll go through it very quickly, but most of this happens from just you reading it and knowing it on your own.
And this is mitosis.
This is mitosis. These are the steps of mitosis. They show you G2. We've kind of already been there. Let's leave G2 and let's go into mitosis, which is this idea that I can distribute my DNA. I've already condensed it down into these things called chromosomes. So I've already got everything packaged up. Prometaphase happens. I'm sorry, let's just do prophase. It's called PMAT. That's the acronym I use. In PMAT, the first one is prophase. You notice in prophase what's happening is the nucleus starts to disintegrate. You can see there's more than just the pores in it starting to break apart. Do you see how your DNA was not condensed down, but now it is condensed down into these almost chromosomes? So you're starting to condense some DNA, some chromatin. You're starting to have a nucleus that's dissolving effectively. And also, we're seeing that these spindle fibers, these aster-forming like centrosomes, are going to opposite ends of the cell. Cool? As that happens, you go into metaphase. I always skip prometaphase. So you got prophase, then you have metaphase. Look at metaphase here. Is there a nucleus left? It's gone. Do you see the chromosomes? They're all lined up with that imaginary equator. That's how you know you're in metaphase, because you have all the chromosomes lined up. Do you see the microtubules, the yellow strings? Do you see where they're attaching to? They're attaching to the centromere. They're about ready to pull the two sister chromatids apart. Then in anaphase, do you see what's happening? You're separating out the sister chromatids. And in telophase, you lead right into cytokinesis and the formation of the two new daughter cells. Your textbook reading, y'all need to be to this picture for reading tomorrow. We'll have a quiz at the start of class. Have a good day.